At MIPS, the Monash Institute of Pharmaceutical Sciences, we were working towards vaccines for influenza. When the current pandemic hit, we were kind of well suited to respond to that.
We were working on several other projects, and then Corona hit. And we suddenly thought, well, this is actually quite a good opportunity to contribute with all the knowledge that we have towards something that will make a huge impact if it works. We already know what we have to do because we've done it before. We're a very small team here within MIPS. It's Harry, me, Colin, and then 2 PhD students. Colin's leadership, having him leading the project, he has great ideas. He can see the bigger picture that we might not necessarily see immediately.
In Australia, Monash has been on the cutting edge on developing new things and new technologies.
A vaccine usually has the pathogen in a specific form. It's a very small fraction of what the pathogen normally is. So the human recognizes it as foreign. It's like, oh, I'm not happy, but you're not getting sick from it. Your body is just primed to make antibodies so that when you're getting attacked by the real pathogen, your body already has made these antibodies. The technology that we are developing here is mRNA, so messenger RNA, and RNA is a bit like DNA. It's not a drug molecule like you would usually take when you have a headache or something. It's a message that the cell will then read to make something, to make a protein. So you administer the message, the body makes a protein and recognizes it as foreign. So it's gonna make antibodies against that protein inside the cell. The cell is not making the whole virus, but what we did instead, we looked at the region in the virus which is mostly responsible for attaching to the host cell. And we teach the cell to make that specific subpart of the virus. Depending then on the pathogen that you want to fight, you would have a different message, but the delivery system that protects the mRNA could be the same.
With mRNA, in our calculations, it's not going to cost a lot because the actual footprint that you need, the size of the room, a classroom size facility, you'll be able to produce it rapidly if you want. We've actually built the vaccine, the actual component, we've built it within 3 weeks. The long part was approval of the trial, the preclinical trials, and then actually doing that because that takes some time. Safety has to be taken very seriously, and in my case, I need to make sure that whatever vaccine goes into people is something that is safe. You're giving to healthy patients, so it's really important for this vaccine to be safe.
That's why clinical trials are really needed. They need to be of a certain length in order to be able to gather enough data. Some drugs have adverse events or adverse reaction that only come after repeated dosage, and you want to capture all of that before you transition onto the market.
Being a pharmacist, being a pharmaceutical scientist, you always want to, have that compass facing towards people. The therapy that can be given and translated to people so it can actually save people's lives.